Tissue and cell culture method for plants and animals. Tissue culture. The tissue culture refers to a method in which fragments of tissue, plants or animal tissue are introduced into a new artificial environment where they continue to function or grow. While fragments of a tissue are often used, it is important to note that entire organs are often used for tissue culture processes. Here, such growth media as broth and agar are used to facilitate the process. Types of tissue culture. 1. Seed culture. The seed culture is a type of tissue culture that is primarily used for plants such as a orchid. For this method, explants tissue from plants are obtained from an in vitro derived plants and introduced into an artificial environment where they get to proliferate. If the plant material is used directly for this purpose, must be sterilized to prevent tissue damage and ensure optimum regeneration. 2. Embryo culture. Embryo culture is a type of tissue culture that involves the isolation of an embryo from a given organism for in vitro growth. The embryo culture may involve the use of a mature embryo, whereas a mature embryo for culture or essentially obtained from ripe seeds. Immature embryo rescue involves the use of immature embryos from unripe hybrid seeds that fail to germinate. In doing so, the embryo is ultimately able to produce a viable plant. For embryo culture, the ovule, seed or fruit from which the embryo is to be obtained is sterilized, and therefore the embryo does not have to be sterilized again. The salt sucrose may be used to provide the embryo with nutrients. The culture is enriched with organic or inorganic compounds, the inorganic salts as well as growth regulator. Callus culture. The callus is produced when explant cells are cultured in an appropriate medium. A good example for this is the tumor tissue that grows out of the wound of differentiated tissue or organ. In practice, the callus culture involves the growth of callus composed of differentiated and non-differentiated cells, which is the following by a procedure that induces organ differentiation. For this type of tissue culture, the culture is often sustained on a gel medium, which is composed of agar and a mixture of given macro and micronutrients depending on the type of the cells. Different types of basal salt mixture such as Mauritius and SCU media are also used in addition to vitamins to enhance the growth. For organ culture. The organ culture is a type of tissue culture that involves isolating an organ for in vitro growth. Here, any organ plants can be used as an explant for the culture process: shoot, root, leaf and flowers. With organ culture, or as is with their various tissue components, the method is used for preserving their structure or functions, which allows the organs to still resemble and retains the characteristics they would have in vivo. Here, new growth differentiated structures continues, giving that the organ retain its physiological feature. As such, an organ helps to provide information on pattern of growth, differentiation as well as development. There are a number of methods that can be used for organ culture. These includes 1. Plasma clot method. Here, the method involves the use of clot that is composed of plasma and chick embryo extract or any other extract in a watch glass. 2. Raft method. For this method, the explant is placed on a raft of lens paper or rayon acetate that floats on a serum in a watch glass. 3. Agar gel method. The medium used for this method is composed of a salt solution, the serum as well as the embryo extract or a mixture of various amino acids and vitamins with 1% of agar. 4. Grid method. The grid method, as the name suggests, involves the use of perforated stainless steel sheet on which the tissue of interest is placed before being placed in a culture chamber containing fluid medium. 5. Protoplast culture. The protoplast cell without cell wall. The protoplast is the term used to refer to cell, fungi, bacteria, plant cells, etc., in which the cell wall has been removed, which is why they are also referred to as a naked cell. Once the protoplast has been regenerated a cell wall, then it goes through the process of cell division to form callus, which may be the subculture for continued growth. The protoplast culture is an important method that provides numerous single cell that can be used for various studies. 6. Some of the other types of tissue culture includes 1. Single cell culture. The basic principles of single cell culture is the isolation of large number of intact living cells and culture them on a suitable nutrient medium for their requisite growth and development. The single cells can be isolated from a variety of tissue and organs of green plants as well as from callus tissue and cell suspension. 2. Suspension culture. The unorganized plant cell can be grown as callus in aggregated tissue masses, or they can be freely dispersed in agitated liquid media. Under agitation, the single cell breaks off and by division forms cell clumps. The tissue and the cell culture is an agitated liquid medium produce a suspension of single cells and cell clumps of few to many cells. These are called suspension culture. 3. Anther culture. Anther culture is a method of androgenesis, that is, production of haploid plantlets by the in vitro condition where the anther is separated from the buds, and it is followed by the sterilization and the culture in nutrient medium to give rise to the haploid plantlets. 4. Pollen culture. The pollen culture, microspore culture, is a technique in which the haploid plants are obtained from isolated pollen grains, while in anther culture, those are obtained from pollens by placing anther on a suitable synthetic culture medium. 5. Somatic embryogenesis. The somatic embryogenesis is a process in which a single or group of somatic cell initiates the development pathway that leads to the formation of non-zygotic embryo. The zygotic embryos developed from the fertilized egg term zygote, whereas non-zygotic embryo develops from the cells other than the zygote. The somatic embryo are bipolar structure in that they have a radical and a plumule. These embryos have no connections with the pre-existing vascular tissue within the maternal callus. The somatic embryo can be produced either directly or indirectly. Major steps of tissue culture plants. Initial phase, stage 1. The initial phase is the first phase of tissue culture. Here the tissue of interest is obtained and introduced and sterilized in order to prevent any microorganisms from negatively affecting the process. It is during this stage that the tissue is initiated into culture. Multiplication phase, stage 2. The multiplication phase is the second step of tissue culture where the in vitro plant materials is redivided and then introduced into the medium. Here the medium is composed of appropriate components for growth, including the regulator and nutrient. These are responsible for proliferation of tissue and the production of multiple shoots. This step is often repeated several times in order to obtain the desired number of plants. Root formation, stage 3. It is at this phase that the roots are formed. Here, hormones are required in order to induce rooting and consequently complete plantlets. The process of plant tissue culture. This process involves the use of small pieces of a given plant of interest. Once the tissue is obtained, it is then cultured in the appropriate medium under sterile condition to prevent various types of microorganisms from affecting the process. The following is general procedure for plant tissue culture. Medium preparation. 1. The appropriate mixture such as the MS mixture is mixed and distilled water and stirred while adding the appropriate amount of sugar and sugar mixture. Here sodium hydroxide and hydrochloric acid is used to adjust the pH. The content used here will depend on the plant to be cultured and the number of the tissue to be cultured. 2. The agar is added to the mixture, heat and stirred to dissolve. 3. After cooling, the warm medium is poured into the polycarbonate tubes to a depth of about 4 cm. 4. While lids sitting on the tubes are placed in a pressure cooker and sterilized for 20 minutes. Growth regulator. The plant growth regulators are composed which at very low concentration can modify growth or plant morphogenesis. There are 7 main classes of plant growth regulator used in plant cell culture, namely 1 auxins, 2 cytokinins, 3 gibberellins, 4 abscisic acid, and 5 ethylene, 6 polyamide, and 7 jasmonic acid. Plant preparation. 1. Cut the plant pot into small pieces. Example, cauliflower can be cut to florets of about 1 centimeter across. On the other hand, such pot of as African violet leaves can be used. 2. Using detergents and water, wash the plant pot for about 20 minutes. 3. Transfer the plant pot into a sterile Clorox solution. Shake for a minute and leave it to soak for 20 minutes. 4. Using a lid, gently discard the Clorox and retain the plant pots in the container and then cap the container. Transferring the plant material to a tissue culture medium. 1. 70% of ethanol should be 70% alcohol should be used for sterilization of equipment used and containers. 2. This procedure will result in the development of calyx, which then produces shoots after a week ahead. Once the shoot develops, then the plant sections may be placed in a right environment, well lit, warmth, etc. for further growth. 4. The plant material should be sterilized to remove any bacteria or spores that may be present. 5. For plants, the medium culture acts as a greenhouse that provides the explant with ideal environment for optimum growth. This includes being free of microorganisms, nutrients, as well as the right balance of chemical and hormones. Such medium as BAP, TDZ are used, while such hormones as IBA and IAA are used to induce growth. Technique for plants in vitro culture. Microporation. This technique is used for the purpose of developing high quality clonal plants. A clone is a group of identical cells. This has the potential to provide rapid and large scale propagation of new genotype. Somatic cell genetics, used for haploid production and somatic hybridization. Transgenic plants. It is used for expression of mammalian genes or plant genes for various species. It have provided beneficial for engineering of species that are resistant against viruses and insects. Methods of plant tissue culture. Plant tissue culture microporation. It is a practice used to propagate plants under sterile condition, often to produce clones of a plant. Different techniques in plant tissue culture may offer certain advantages over traditional method of propagation, including 1. Production of exact copies of plants that produces particularly good flowers. Methods of plant tissue culture. Plant tissue culture micropropagation. It is the practice used to propagate plants under sterile condition, often to produce clones of plants. Different techniques in plant tissue culture may offer certain advantages over traditional method of propagation, including 1. Production of exact copies of plants and that produces particularly good flowers, fruits, or they have other desirable traits. 2. To quickly produces mature plants. 3. The production of multiples of plants in absence of seeds or necessary pollinators to produce seeds. The regeneration of whole plants forms plant cells that have genetically modified. The production of plants in sterile containers that allows them to move with a greatly reduced chance of transmitting disease, pests, and pathogens. The production of plants from seeds that otherwise have very low chance of germinating and growing, that is orchid and nepenthes. To clean plants of viral other infections and to quickly multiply these plants as clean stock for horticulture and agriculture. Stages of microporation. There are 3 major ways by which microporation can be achieved. These are enhancing axillary bud breaking, 2. Production of adventitious bud directly or indirectly on explant. 3. Somatic embryogenesis. The plant microporation in vitro is an integrated process by which the cells, tissue, or organs of selected plants are isolated surface sterilized and incubated in growth promoting sterile environment to produce many clonal plantlets. The physical and the chemical environment factor affects the initiation of culture, the growth, behavior, and regeneration. The 5 operational stages are involved, 3 of them in vitro, while one stage occurs in the greenhouse environment. This classification of production stage has been suggested by DeBerg and Manny in 1981. Stage 0 explant source and mother plant. Stage 1 explant establishment in culture. Stage 2 rapid multiplication. Stage 3 plantlet establishment, elongation and rooting. And stage 4 acclimatization, application of micropropagation. Micropropagation offers significant advantages in quality, quantity, and economics over conventional method of vegetative propagation for many species. Techniques for micropropagation: 1. Plant cell culture. It was Haberland in 1890 which put forth the pioneering attempt to culture mechanically isolated mesophil cells. He succeeded in maintaining these cells alive for 10 days in culture, but it did not divide. He failed to achieve divisions of isolated cells. Since then, the process in the field of isolating single cell but culturing individual cells has been spectacular that it has become possible not only to culture isolated single cell but also induce division in such cells in complete isolation, and finally to regenerate whole plant from them. The most important aspects that deserves attention in the isolation cell culture technology is the initiation of division and proliferation of isolated single cells, which starts only when they are placed into conditions medium. The isolated single plant cell resembles with the animal cell in this respect. Application of cell culture. 1. Resistance to disease. Tobacco protoplast selected from resistance to methionine sulfoxamine showed enhanced resistance to Pseudomonas tabaci. 2. Resistance to herbicide. In the recent past, the fertile plant resistance to herbicide such as amitrol 2,4-D, sodium chlorate, picloram, chlorsulfuron, and sulfomethurone methyl has been selected from cultivated cell of Nicotiana. 3. Resistance to amino acids and their analogues. A potato cell line resistant to 5-methyltryptophan has been used to demonstrate the presence of 2 possible isoenzymes of anthranilate synthetase, the first regulatory enzyme unique to tryptophan biosynthesis. 4. Secondary metabolic productions. A few but well-known established enzymes of secondary metabolic production is high amount by selecting cell lines or glutathione and nicotine from Nicotiana tabacum, anthraquinone from Morinda cotrifolia, rosmarinic acid from Coleus blumi, ajmalicin and serpentine from Catranthus roseus, diogenin from Dioscloria deltoidea. 5. Biotransformation means the conversion of less useful compound into more useful compounds by plant cells. The biotransformation of digitoxin or beta methyl digitoxin to digitoxin or beta methyl digitoxin by specific hydroxylation of suspension culture of digitalis lantana can carry out biotransformation at 15% conservation in 24 hours and 70% in 7 days. Digoxin is used in cardiac treatment. The cell culture of stevia rebondiana and digitalis purpura can biotransform the steviol to steviol biocide and stevioside. 2. Callus culture. The callus is a amorphous mass of unorganized thin-walled pharyngeal cells. When a plant is wounded, callus formation occurs at the cut surface, and it is thought to be protective response by the plant to seal off damaged tissue. In culture, callus is initiated by placing a fragment of plant tissue explant on solid culture medium under aseptic condition. The callus is induced and formed from the proliferating cells at the cut surface of explant tissue. Depending upon the species, the callus can be initiated from a variety of tissue employing the appropriate growth medium. However, the rapid cell division can be more easily induced in some tissue than the other. The in vitro formation and the proliferation of callus is enhanced by the presence in the medium of hormones or auxins and cytokinins that promotes cell division and elongation. During the in vitro initiation of callus, the differentiation and specialization that occurs in the parent plant is reversed and the cell of plants become de-differentiated. The process of de-differentiation is characterized by the changes in the metabolic activity, the appearance of storage products and rapid cell division that give rise to undifferentiated and unorganized parenchyma cells. Somatic embryogenesis. The somatic embryogenesis refers to the process in which the somatic or non-sexual cells are induced to form bipolar embryos through a series of developmental steps like throughs occurring during in vivo embryogenesis. The somatic embryo formation has been achieved for a variety of plant species including gymnosperm. The somatic somatic angiosperms and embryogenesis is different from organogenesis in that regeneration and organization or bipolar. The shoots and roots meristems are formed simultaneously from group of the cells embryonic masses. As with the organogenesis, the differentiation, morphogenesis, and organization of somatic embryo takes place directly in the explant or from the callus. The somatic embryogenesis is often favored by culture in an agitated liquid medium. Advantages of somatic embryogenesis. 1. Rapid multiplication through productions of somatic embryogenesis in cell culture and use of bioreactors for scale-up industries. Thus, mass propagation of plant can be performed. 2. Somatic embryogenesis growth individually makes the system easy to manipulate to subculture and to make transgenic plants. 3. Provide an important source for analysis of molecular and biochemical event that occurs during the induction and maturation of embryo. 4. Synthetic seed, artificial seed, and somatic seed. The synthetic seed or artificially encapsulated somatic embryo, shoot buds, cell aggregates, or any other tissue that can be used for sowing as a seed and which possesses the capability to convert into plant under in vitro conditions. 1. Hydrated synthetic seed consists of somatic embryo individually encapsulated in hydrogel such as calcium alginate. 2. Desiccate synthetic seeds are produced by coating somatic embryos in polyoxyethylene glycol. Method of production. Following steps are followed for production of synthetic seeds. 1. Induction of somatic embryo from cell suspension or callus culture. 2. Mixing the embryo with 2% of sodium alginate. 3. Immersion of embryo in the bath of calcium salt, example solution of calcium chloride or calcium nitrate for 30 minutes. This leads to very quick complex formation at the surface due to the exchange of ion, example sodium and Na plus and Ca2 plus. As a result, the individual embryos are enclosed in clear and hardened beads of about 4 millimeter. The hardening of calcium alginate is modulated with the concentration of sodium alginate along with the duration of complexing. Usually 30 to 100 millimolar Ca2 plus is used for sieving the beads through the nylon mesh. Calcium NaNO3 twice calcium nitrate solution is recycled. And 5. Testing the growth vigor of beads by plating in sand or soil amended with pesticides. Advantages of artificial seed: 1. Reduces cost of transportation. 2. Production of large number of identical embryos. 3. Germ plast conservation through cryopreservation. 4. Study of seed coat formation is possible. 5. Useful in tree genetic engineering. 6. Production of synthetic seeds is possible in any time, in any season of year. 7. The problem of dormancy can be avoided, thereby shortening the life cycle of plant. 5. Haploidy. Haploidy plants have the gametophytic number of chromosomes, a single set N chromosome in the sporophyte. The haploid cell or individual are those in which the original chromosome number has been reduced by the half. These are the immense importance for plant improvement, useful in mutation and for production of homozygous diploid plants. In theory, haploid plants can be produced by the in vitro cell of haploid gametophytic cells, that is unpollinated ovaries or ovule and anthers or pollen. The viable haploid plants have been produced by ovary and ovule culture for some crops. The procedure are, however, technically laborious and yield low. There are two in vitro procedures that are now routinely used in the production of haploid. 1. Anther culture. 2. Culture of isolated microspores. The development of haploid plants from the male gametophytic tissue is known as androgenesis. Anther culture. It is a rapid and relatively simple procedure by which the pollen producing anther are collected from the flower at a certain step of development. Sometimes they are given a low or high temperature pre-treatment before sterilization, a culturing on a specific nutrient medium. The mature pollen, the male gametophyte, develops from haploid microspores that are in turn formed by meiosis in pollen mother cells. The microspores have a single nucleus and mature pollen 3 nuclei. When the anther continuing the microspore at the middle to late unnucleated stage or cultured on the solid medium, the individual microspores forms the callus. The plants with half of the chromosome number can be regenerated from the callus directly by embryogenesis or indirectly by organogenesis. Alternatively, the microspores may give rise to embryoids, the aggregates of cells with recognized embryonic structure that are capable of differentiation of embryo. B. Microspore culture. It involves the physical isolation of a microspore from the anther at a specific stage of development. After a suitable pre-treatment, the anther are homogenized and microspore are isolated and purified from the homogenated by filtration and centrifugation step. The purified microspores are then cultured in the liquid medium. The small embryos develop directly from the microspore embryogenesis and are transferred to a regenerative medium. In microspore culture, problems of anther culture such as mixoploid types, mixoploidy and albino plants in the regenerated plants are less frequent. The microspore cultures are the ideal material for genetic transformation. Advantages of haploid culture are: 1. To produce virus-free plant. 2. In-plant breeding to obtain pure line varieties. 3. Mutation breeding. 4. Tree breeding. 5. Development of new varieties. Mingo, the first licensed cultivar of barley, was produced using the haploid. 6. Somaclonal variations. When the plants are grown in culture, much genomic varieties is produced. This variability can be found in callus and suspension culture, tissue culture. protoplast culture, and other in vitro culture system. Such variations observed among the plants regenerated from tissue culture are called somoclonal variations. Scowcroft 1985. The frequent genetic modifications are known to occur during the process of cell or tissue culture. These modification tends to be inherent as mutation among the progeny of regenerated plants. The source of explant is a critical factor or variable for somoclonal variations. The plant cell, tissue, and somatic tissue developed from the various explants are source for generating the somoclonal variations. The somoclonal variations has been reported in number of species such as sugarcane, potato, rice, brassica, tobacco, tomato, and other various agronomic traits like disease resistance, plant height, maturity, and for many physiological and biochemical traits. Applications: increase genetic variability for agronomic trait. 2. In vitro selection has been selected desirable somoclones including tolerance to pathotoxins, herbicides, and disease. Example: tobacco variants resistance to Pseudomonas syringae and Alternaria alternae have been obtained. 3. Variants with improved traits have been obtained, giving rise to a new useful germplasm as well as cultivar. Example: tobacco germplasm having resistance to potato virus Y, and rice dwarf cultivar with 10% high yield and tolerance to salt have been obtained. 7. Protoplast fusion and somatic hybridization. The plant cell is a multi-layered structure composed of a polysaccharide and protein. Careful removal of plant cell wall results in a viable spherical cell called protoplast. The protoplast contains original cells bounded by the plasma membrane. With the appropriate culture conditions, protoplast will resynthesize the cell wall and undergo cell divisions to form callus, from which new plants can regenerate. The protoplast can be isolated from varieties of whole plant tissue and from plant tissue cultures like callus and suspension culture. The removal of the wall for protoplast is achieved either by mechanical means, removal of cell wall by ultraknife of plasmolyzed cell, or enzymatic digestion, which includes the combination of cellulase, hemicellulase, and pectinase. In plant breeding, the technique of somatic hybridization by protoplast fusion is particularly useful way of introducing the genetic variations from distant relative into crop plants in circumstances where sexual hybridization is prevented by reproductive barriers. When two or more protoplasts fuse to form a new cell, the parent nuclei may remain separate or fuse to form a somatic hybrid. If one of the nuclei is lost after fusion, the cytoplasm of two parent protoplasts will still coalesce and form what is known as a cytoplasmic hybrid or cybrid. The isolated protoplast can be fused spontaneously, although this occurs infrequently because of mutational repulsion that results from the net negative surface charge or protoplast plasma membrane. Nevertheless, fusion can be induced either by application of compounds known as fusogenic agent. Example: calcium nitrate, PEG, high pH, and a calcium ion dextran, or by placing the protoplast in an electric field, electrofusion, or electroporation. The fusogenic agents that tends to either neutralize or shield the surface charge so that adjacent protoplasts come in proximity, adhere, and coalesce. The fusion is induced by high concentration of calcium ion at high pH 10, and high temperature 370 degrees centigrade. However, the most widely used fusogenic agent is polyethylene glycol (PEG). The electrofusion takes place in two stages. First, the protoplast suspension is placed in a fusion cell between two metal electrodes and subjected to a non-uniform altering electric field. The protoplast membrane becomes differentially charge and positive region or attracted to negative region on adjacent protoplast resulting ion the protoplast alliging in chain. In the second step, the high voltage DC supply is applied to the align protoplast that causes the breakdown of adjacent membrane and fusion. The flow cytometer can be used to identify the fusion products by cell sorting and estimation of nuclear DNA. Advantages of somatic hybridization. This technique can be used as an alternative to obtain tetraploid and amphidiploid when mitotic doubling is impossible. 2. It allows genetic recombination of sexually incompatible genotype, which is impossible using conventional technique of hybridization. 3. It helps to produce hybrids between the species that are too distinct to be hybridized sexually. 4. Both parental species contribute equally for hybrid in somatic hybridization. 5. The protoplast can be pre-treated prior to fusion with increased gene integration. 6. The wild relative of a cultivated variety having dominant and recessive genes can be exploited to improve crop yield. For instance, the wild relative of a cultivated sweet potato contains the dominant and resistant gene against against root knot nematodes. 7. It can be employed in the improvement of plants wherein sexual reproduction it is either difficult or absent, such as banana, cassava, potato, sugarcane, and yam. Here, the fusion derived plants need not be fertile, circumventing the problem of infertility. 8. Hybrids. These are obtained when a nucleus is derived from one parent and a cytoplasm is derived from both the parents. The cytoplasmic hybrids can be obtained by using any of the following methods. A. Fusion of normal protoplast from one parent with eunucleated protoplast from other parent. B. The fusion of normal protoplast from one parent and a protoplast containing non-viable nuclei from the other parent. C. Selective elimination of one of the nuclei from the heterokaryon. D. Selective elimination of chromosomes of one parent at the later stage after fusion of nuclei. The hybrids have been produced in many cases mainly to transfer cytoplasmic organelles. The hybridization have been successfully employed to make intergeneric and interspecific transfer of cytoplasm in tobacco, petunia, rice, and brassica species. Animal cell culture. The animal cell are more difficult to culture than microorganisms because they require many more nutrient and typically grown only when attached to specially coated surface. Despite these difficulties, various kinds of animal cells, including both undifferentiated and differentiated ones, can be cultured successfully. Nutritional requirement. The rich medium are required for culture of animals. 9 amino acids, referred to as the essential amino acids, cannot be synthesized by adult vertebrate animals and thus must be obtained from their diet. The animal cells grow in culture also must be supplied with these 9 amino acids, namely histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. In addition, most cultured cell requires cysteine, glutamine, and tyrosine. In the intact animals, these 3 amino acids are synthesized by specialized cells. For example, liver cells may make tyrosine from phenylalanine, and both liver and kidney cells can make glutamine. The animal cells, both within the organisms and in the culture, can synthesize the 8 remaining amino acids. Thus, these amino acids need not be present in the diet or culture medium. The other essential components of a medium for culturing animal cells are vitamins, which the cells cannot make at all or in aqueous adequate amount. The various salts, sugar, and serum, the non-cellular part of the blood. The nutrient medium used for culture of animal cells and the tissue must be able to support their survival as well as growth. That is, must provide the nutritional, hormonal, and stromal factor. The various types of media used for tissue culture may be grouped into two broad categories: one natural medium and two artificial medium. The choice of the media depends mainly on the type of the cell to be cultured: normal, immortalized, and transformed, and objectives of the culture: growth, survival, differentiation, production of desired protein. Natural medium. These medium consists solely of naturally occurring biological fluid and or of the following three types. A. Clots. The most commonly used clots are plasma clots, which have been in use for a long time. B. Biological fluid. Of various biological fluid used as a culture medium. Example: amniotic fluid, ascitic and pleural fluid, aqueous humor from eye, insect, hemolymph, serum. The serum is most widely used. C. Tissue extract. The chick embryo extract is the most commonly used tissue extract, but bovine embryo extract is also used. Artificial medium. The different artificial medium has been devised to serve one of the following purposes: one, immediate survival. A balanced salt solution with a specified pH and osmotic pressure is adequate. Two, prolonged survival. A balanced salt solution supplemented with serum or with suitable formulation of organic compounds. Three, indefined growth. And four, specialized function. The various artificial medium developed for cell culture may be grouped into four classes: one, serum containing medium; two, serum free medium; three, chemically defined medium; and four, protein free medium. Primary cell culture. The primary culture is the stage of culture following isolation of cells, but before the first subculture there are three stages of to be considered: one, isolation of tissue through dissection and or disaggregation; and three, culture following seeding into the culture vessel. Following isolation, the primary cell culture may be obtained either by allowing the cell to migrate out from fragments of tissue adhering to a suitable substrate, or by disaggregating the tissue mechanically or enzymatically to produce a suspension of cells, some of which will ultimately attach to the substrate. It appears to be essential for most normal untransformed cell except for hematopoietic cells attached to a flat surface in order to survive and proliferate with maximum efficiency. The transformed cells, on other hand, particularly cells from transplantable animal tumor, are often able to proliferate in suspension. Isolation of tissue. On dissection, selection of required tissue, removal of fatty and necrotic tissue, and is divided into three types: fine dissection, chopping down to explant size, and mechanical disaggregation, example sieving, syringe, or vigorous pipetting; and then third is enzymatic disaggregation. In fine dissection, primary explant, and micro mechanical disaggregation. It's been to dispersed primary culture, and enzymatic disaggregation divided into three types: cold trypsin, warm trypsin, and collagenase. And the primary explant is divided into explant and outgrowth. The mechanical, the explant is divided into transfer. It's been transfer, and it's for second explant culture. And outgrowth is been used for subculture, and this dispersed primary culture on subculture to into cell line. The cold trypsin in enzymatic disaggregation: cold trypsin overnight cold short incubation, and warm trypsin long incubation repeated sampling, and collagenase long incubation complete medium. The old night cold short incubation is been centrifuged, and long incubation repeat samples is been centrifuged, and long incubation complete medium is been centrifuged, and it has been resuspended and seed on the primary dispensed in the primary culture, and it is in subculture, and it's been cell lined. The enzymes used most frequently are crude preparations of trypsin, collagenase, elastase, hyaluronidase, DNase, pronase, dispase alone or in various combination. The crude preparation are often more successful than purified enzyme preparation, as the former contains other proteases as contain, although the latter are generally less toxic and more specific interaction. The trypsin and pronase gives the most complete disaggregation but may damage the cells. The collagenase and the dispase, on the other hand, gives incomplete disaggregation but are less harmful. Hyaluronidase can be used in conjugation with collagenase to digest the intracellular matrix, and DNA is employed to disperse DNA release from lysed cell, as it tends to impair proteolysis and promote reaggregation. Techniques for mass culture of animal cell line. The cultivation of animal cell differs significantly from the technique used with bacteria, yeast, and fungi. The tissue excised from specific organs of animals such as lungs, kidney, under aseptic conditions are transferred into the growth medium containing serum and a small amount of antibiotic in a small tea flask. These cells forms a primary culture. Unlike plant cells, the primary mammalian cells do not normally form aggregate but grow in the form of culture. Unlike plant cells, the primary mammalian cells do not normally form aggregate but grow in the form of a monolayer on support surface, such as glass surfaces of flask. Using the proteolytic enzyme trypsin, the individual cells in the tissue can be separated to form a single cell culture. The cell directly derived from excised tissue are known as a primary culture. A cell line obtained from the primary culture is known as a secondary culture. The cells are removed from the surface of the flask using the solution of EDTA, trypsin, collagenase, or pronase. The exposure time of the cell for cell removal is 5 to 30 minutes at 37 degrees centigrade. After cell are removed from the surface, the serum is added to the cell culture bottle. The serum containing suspension is centrifuged and washed, and with buffer isotonic saline solution are used to inoculate secondary culture. The culture prepared directly from the tissue of an organism, that is without cell proliferation in vitro, or called primary culture, then can be made with or without an initial fractionation step to separate different cell types. In many cases, the cell in primary culture can be removed from the culture dish and made to proliferate to form many so-called secondary culture. In this way, they may be repeatedly subculture for weeks or months. Such cells often display many of the different properties appropriate to their origin. The fibroblasts continue to secrete collagen. The cell derived from embryonic skeletal muscle fused to form muscles fibers that contract spontaneously in the culture dish. The nerve cells extend to auxins that are electrically excitable and make synapses with other nerve cells, and epithelial cells forms extensive sheet with many of the properties of an intact epithelium. Because these phenomena occurs in culture, they are accessible to study in way that are often not possible in intact tissue. To cell lines. To be able to clone individual cell, modify cell behavior, or selective mutant, biologists often wants to maintain cell culture for many more than 100 doublings. This is possible with the cell derived from some tumors and with rare cell that arises spontaneously because they have undergone genetic changes that endow them with the ability to grow indefinitely. The genetic changes that allows these cells to grow indefinitely are collectively called as oncogenic transformation, and the cells are said to be oncogenic transformed or simple transformed. A culture of cells with an indefinite lifespan is considered immortal. Such a culture are called a cell line. To distinguish it from an impermanent cell strain. Stages in establishing of cell culture. When an initial explant made of human cell, some cells die and other, mainly fibroblasts, starts to grow. The growth rate increases in the phase 1. In the remaining cells are continuously diluted. The cell strain grows at a constant rate for about 50 cell generation, phase 2, after which the growth rate falls rapidly. During the ensuing period of increasing cell death in the phase 3, all the cells in the culture eventually die. In the in a culture prepared for from mouse or other rodent embryo cells, there is a initial cell death couples with the emerging of healthy growing cells. As these are diluted and allowed to continue growth, they soon begins to lose growth and potential, and most cell dies. The culture goes into crisis. Very rare cells do not die but continue growing until their progeny overgrow the culture. These cells constitute a cell line, which will grow forever if it is appropriately diluted and fed with a nutrient. The cells are immortal. The ability of the culture cell to grow indefinitely, or their tendency to transform, varies depending on the animal species from which the cell originates. The normal chicken rarely are transformed and die out after only a few doubling. Even tumor cells from chickens almost never exhibit immortality. Among human cells, only tumor cell grows indefinitely. The HeLa cells, the first human cell type to be grown in culture, was originally obtained in 1952 from a malignant tumor carcinoma of uterine cervixes. This cell line have been unavailable for research on human cells. 3. Bioreactor, a consideration for animal cell culture. The mammalian cells are large 10 to 20 micrometer diameter, slow growing TD 10 to 50 H, and very shear sensitive. Moreover, some animal cells are anchorage dependent and much grow on surface of the glass, especially treated plastics, natural polymers such as collagen and or other support material. Some are not anchorage depending and can grow in suspension culture. The product concentration titer is usually very low microgram per ml, and the toxic metabolites such as ammonium and lactate are produced during the growth. These properties of animal cell set certain constraints on design of a animal cell bioreactor. 4. Monolayer culture. The monolayer culture are essential for anchorage dependent cell. The scaling of such culture is based on increasing the availability surface by using plates, spirals, ceramics, micro carriers more effective. Application of animal cell culture. The animal cell culture are used to produce virus vaccine as well as varieties of useful biochemical, which are mainly high molecular weight proteins like enzyme, hormone, cellular biochemical like interferons and immunobiological compounds including monoclonal antibodies. The animal cells are also good host for the expression of recombinant DNA. The molecules and few commercial products have been or been developed. Initially, where virus vaccines were dominant commercial products from the cell culture, but at present monoclonal antibody production is the chief commercial activity. It is expected that the recombinant protein would becomes the primary product from the cell culture soon. The transplantable tissues and organs are another very valuable product from the cell culture. The artificial skin are already in use for grafting in burn and other patients, and efforts are focused on developing transplantable cartilage and other tissues. The animal cell products usually consist of high molecular weight proteins with or without a glycosidic group. A few enzyme, hormones, vaccines, immunobiological monoclonal antibody, lymphokines, and anti-cancer agents can be produced using animal cell culture technology. The major type of immunoglobulin product produced by the animal cells are monoclonal antibody, immunoglobulin, regulator, interleukin, and lymphokines, and 3 virus vaccine, prophylactics. One, monoclonal antibody. One of the most important products of animal cell culture has been monoclonal antibodies, MBS. The monoclonal antibodies are produced by the hybridomo cells and are used in the diagnostic assay system for therapeutic purposes and for biological separation, for example, affinity chromatography. The monoclonal antibodies have been used to determine the well over 100 drugs, toxins, vitamins and other biological components as a diagnostic agent. The monoclonal antibodies are also used for the chromatographic separation to purify protein molecules. 2. Immunoglobulin regulator. The interferons and anti-canceral glycoprotein secreted by the animal cell upon exposure to cancer-causing agent is an example of a immunoregulator produced by the mammalian cells. The interferon can be produced by either animal cell or recombinant genetically engineered bacteria. Other immunoregulator or lymphokines, hormone protein regulating immune response of a human body, interleukins, anti-cancer agents, tissue plasminogens activator, a compound preventing blood clotting, and the thymosin. The production of immunoregulator is a very compromising area with a great potential for growth soon. The list of products generated likely to be generated from the cultured animal cells: virus vaccine, cancer, distemper, saline dog kidney, foot and mouth disease (FMD), cow kidney, saline, hepatitis B, virus vaccine, saline, human plasma derived vaccine, virus vaccine, influenza, chick embryo, measles, chick embryo, mumps, chick embryo, polio, primate kidney, rabies in chicken in human diploid cells, and rubella, German measles, MMR in human diploid cells. The remarks from blood of infected individuals, first vaccine from cell culture, and cellular biochemicals, angiogenic factor, human tumor, beta interferon, mouse fibroblast, beta and gamma interferons, Chinese hamster ovary, CHO cell lines, and alpha interferons, human leukocytes. Immunoregulatory biochemicals, interleukin 2, lymphoblastoid T cells, and in continuous cell line. Product enzyme, collagenase, pepsin, renin, trypsin, urokinase, plasminogen activator, cell line, human kidney, tissue type plasminogen activator, cell line, CHO cell line, single cell urokinase type human kidney transform, plasminogen activator, cell line, TCL 598, trypsinogen hydrolase. Hormones, luteinizing hormone, follicle stimulating hormone, chorionic hormone, erythropoietin growth hormone, ACTH, adrenocorticotropic hormone, immunoglobulins, monoclonal antibodies, cell lines, hybridoma clone, and remarks most dominant activity. 3. Virus vaccine. The various virus vaccine prophylactic have been produced for animals and human use. The table summarizes human vaccine produced by mammalian cells. The virus vaccine should provide a lifelong immunity, be safe, and be given as single dose. The animal cell viruses products for human uses: mumps, measles, prospective hepatitis, future common cold, rubella, prospective hepatitis B, future some cancer, rabies, hepatitis B, HSV, rabies, HSV, simplex virus type 1, future AIDS, yellow fever, herpes simplex virus type 2, polio virus, varicella zoster, influenza virus, prospective respiratory syncytial virus, adenovirus, prospective Rift Valley fever, and smallpox virus. 4. Hormones. Some animal hormones are large molecules, 50 to 200 amino acids, and are glycosylated. These hormones can be produced by using cell culture of hormone synthesizing organ. Some potential hormone products from animal cells are follicle stimulating hormones, chorionic hormones, erythropoietin. Some animal hormones are relatively small polypeptide, 20 to 30 amino acids, and may be produced by chemical synthesis. The erythropoietin is a very successful commercial product useful in treating anemia in a wide range of disorders, from patients on artificial kidney to those with AIDS. 5. Enzymes. A few enzymes can be produced by animal cell culture. The synthesis and excretion of these enzymes are a target of genetic engineering. The glycosylation, post-translation modification, excretion from the host organism or potential problem. Some potential enzyme products from animal cell culture are urodinase, renin, asparginase, collagenase, pepsin, trypsin, hyaluronidase, and blood clotting compounds such as factor 7, factor 8, and factor 10. The the large scale production of tissue plasminogen activator is important commercial process. 6. Insecticide. Animal cell culture have been used to produce some insect viruses that are highly specific and safe to environment. Several of the baculoviruses have federal approaches for use as a insecticide. Genetically engineered variant with greater virulence can now be produced. The commercial production of such viruses from issue culture has not yet been accompanied. 7. The whole cell and tissue culture. The production of a differentiated cell for medical use is under early development. The artificial skin for burn patient is one example. The artificial organ and semi-synthetic bone and dental structures are potentially feasible. Objective type questions. Name the organisms on which first cell line was observed. Mouse. The first cell line was observed was mouse fibroblast L cell, which was derived from connective tissue of mouse by exposing it to chemical carcinogen. 2. The agglutination reaction is more sensitive than precipitation for the detection of antigen. The growth of plant tissue in artificial media is called the plant tissue culture. The plant tissue culture is a technique of growing roots, shoot tissue, and organs artificially in an aseptical medium which contains all the essential nutrients for growth. Name the type of culture which is prepared by inoculating directly from tissue of an organism to culture medium. A. Primary cell culture. The primary cell culture is the direct inoculation of tissue of an organism into a culture medium without growing prior to cell proliferation. The dwarf P mutant IE used by Mendel was defective in which of the following enzyme involved in the gibberellin biosynthesis? GA2 beta hydroxylase. The gibberellin is involved in the treatment of a dwarf stem. 3 beta hydroxylation results in the conversion of a C19 gas GA20 and GA4 respectively. In the final step in the formation of a physiological active GAS, there is now increased evidence that in common with the GA20 oxidase, certain GA3 beta hydroxylase may be multifunctional. An enzyme purified from developing embryo of P. vulgaris catalyzed 2,3 desaturation and 2 beta hydroxylation reactions in addition to 3 beta hydroxylation. Indirect immunofluorescence involves fluorescence labeled Immunofluorescence. Immunofluorescence is used to localize the antigen to specific cell types or subcellular components. The assay can also be adapted for the detection and quantification of antibody. The most convenient method is to use antibody generated in different species, specific secondary antibody conjugated with different fluorochromes. Indirect immunofluorescence involves addition of fluorescinated antigen Ig. Among the existing technologies, which are the following vector system which you prefer to use for generating a library for 140 kilobase eukaryotic genome DNA fragment while giving due consideration to size as well as stability of interest? Yeast artificial chromosome are most sophisticated yeast vectors and represents the largest capacity vector available. It is used to clone DNA fragments larger than the 100 kilobase and up to 300 kilobase. Hence, both bacterial artificial chromosome and yeast artificial chromosome may be used to insert 140 kilobase size. However, yeast artificial chromosomes are significantly less stable than bacterial artificial chromosome. Hence, the stable vector to insert DNA of 140 kilobase is bacterial artificial chromosomes. The maximum size of DNA insert that can effectively clone in various E. coli vectors. Vector M13 size in kb of DNA insert 3 kb. Remarks 1.5 kb is the most common. Vector plasmid 8 kb. Remarks most plasmid. Phagmid vector lambda vector 10 kb. Remarks provide the facility of M13 vector as well as lambda GT10 8 kb insert vector. Remarks insertion vector lambda ZAP11 10 kb insertion vector. Remarks lambda EMBIA 20 kb maximum replacement vector. Lambda GEM11 lambda GEM12 23 kb insert of DNA insert maximum replacement vector. Cosmid vector size 40 kb remarks some cosmid. The bacterial artificial chromosome BACS size of DNA insert maximum 300 kb based on E. coli F plasmid. P1 derived artificial chromosome PAC is maximum 300 kb based on E. coli bacteriophage P1. Which of the following is the best for checking mycoplasma contamination in a mammalian cell line? B. ELISA. The mycoplasma detection kit is an ELISA that combines specifically a high sensitivity with a short assay time and easy evaluation. The kit detects the most common mycoplasma achloplasma species. The mycoplasma arginini, mycoplasma hierohinis, mycoplasma orally, A. Laidlavi that contaminate mammalian cell culture. The determination of each species is separately done. The polyclonal antibody each recognizes one of the four most common mycoplasma achloplasma species are coated to a well of microplate. The sample material is added to the well. The mycoplasma compounds contained in infected sample bind to the antibodies specific for the respective mycoplasma achloplasma species. In the next step, the biotin labeled detection antibody are added, then bound to the mycoplasma. A streptavidin alkaline phosphate AP conjugate is added, then bound to the biotin labeled antibody. Next, an AP substrate alkaline phosphatase streptavidin alkaline phosphatase is added and metabolized by the enzyme. The color reaction product is quantified using the microplate ELISA reader. 4. Transgene